Thank you very much for the nice welcome. So I'm going to present to you today, I'm going to give you a bit of an introduction about some of the things that we've been working on, and then I'm going to present a draft of a paper that I haven't presented before, and I would be interested in your comments. And that's where we link the socio-demographic and the genetic predictors to look at childlessness. So I'll talk about that in more detail towards the middle to the end of the presentation. So I think, I've been located in Europe since 1996. I'm Canadian and Dutch nationality. And what I noticed in Europe during that time, and there was a huge growth in childlessness. So this low fertility, or the lowest low fertility, has been a topic of great interest in Europe. So we know, for example, let's see where I'm putting this. Yeah, so countries in the Mediterranean regions, but also in the German-speaking regions, have high levels of childlessness now. For recent cohorts, that about 1 in 4 are actually childless. So this became a very interesting topic to start looking at. We were seeing very low fertility levels and high levels of childlessness. Something else we saw, which I'm assuming this is a group that's familiar with demography and fertility, but what we saw is this huge, if we compare and it's even gone up over time, 1970 to when women's mean age at first birth was compared to more recent times, we see this dramatic shift of about 4 to 6 years. Over time. And biologically, this is really interesting. So when women and men, and particularly women, are having their first child at the age of 29 or 30, which they're doing in many countries in Europe, they're starting to have their children, they're starting their families, actually, when their fertility is starting to decline, or their fecundability, or their ability to conceive. So this was also something that we thought was interesting to look into. So together with Ron Rinfest, but also others, and Francesco and Nicoletta, we did several reviews that we published to look at sort of the socio-demographic explanations. And I don't think this audience is, of course, familiar with these things, but it's things related to the second demographic transition, the availability of contraceptives. We also have the increase in women's higher education and labor market, problems with work-life reconciliation, inequality. All of these things are related to why we probably have these postponement of fertility, but also the rise in childlessness. So for most of my career, I looked at these factors, these socio-demographic factors. And then I went out for dinner with a biologist, a friend of mine, and he started making fun of me, and he was introducing, and we were having some drinks, and he said, "Oh, this is Melinda. You're going to find this really funny. She looks at fertility, but she thinks it doesn't have a biological component." And I mean, everybody thought that was quite funny. And if you look at the social science literature, and indeed what we did, actually, with some of the reviews we've looked at it, but actually, biology and genetics has been largely ignored. So I was trying in this paper to bring together 3 sort of parallel research streams. So what I've often looked at in demography and sociology, these sort of social factors, with some medical factors. So there's a lot of research into infertility related to sperm defects or endometriosis. And then also more recently, and I know you have Dalton Conley here, so you're familiar with the genetic research that's been looking at this as well. So we know, this is probably hard to read, especially for those in the back, but these are the twin studies to date. So we did a summary of twin studies to date looking at the genetic factors, or how heritable age at first birth and number of children ever born is. And I mean, what you find, a summary from these studies, is that there's a huge variation in heritability across the different cohorts, and I'll return to that at the end. So, it's saying that there probably is some genetic component related to age at first birth and number of children ever born. It could be about 25 to 50, and it differs by country and birth cohort. And what's really interesting is, I was just talking with Charles Westhoff about this as well, too. There's virtually no studies of men, so that was something that we really wanted to look into as well. So we think, okay, there could be a genetic basis to fertility. So these are sort of the genetic approaches that we looked at, and I saw that you had Dan Benjamin and David Cesarini earlier, some of these may be approaches that you already know. But we first sort of transcended and didn't look just at twin models, but we used these Gremel models. And basically that's looking at the genetic relatedness of individuals within your data set. So you can move from looking at twins to looking at many individuals within a data set. So we might be 20% related, or we might be 40% related, and then you compare those individuals and that genetic relatedness. And then I'm going to talk to you and show you, I led a GWAS, a genome-wide association study, looking at age at first birth and number of children ever born. And what you can take from these studies is where you isolate actual genes, you create polygenic scores. So this is kind of the glossary of some of the terms I'll be using. And these scores are individual genetic loci, and I'll talk about those in a minute, where you aggregate and weight them and you use them as variables in your models. So that's some of the terminology and the techniques that I'm going to show you that we're using. And if you're interested, we sort of unpack this evolution from sort of classic designs to twin and family designs and Gremel models to using these genetic scores in an article, and I think in a more accessible way if you're interested. So we first started using these Gremel techniques. We looked at twin models as well, but it's always kind of frustrating when you say, okay, yeah, this is 20% heritable or 30% heritable, and you're sitting there, okay, well, great, why? So then we thought, well, it's interesting then to move to look at actual whole genome data and using these Gremel techniques. And as I said, you look at people and you estimate their genetic relatedness between unrelated individuals. The things that I often look at are age at first birth, so tempo and quantum, and number of children ever born. And I'll look at childlessness as well towards the end of the talk. We have to call it human reproductive behavior because we're in an interdisciplinary environment. And fertility, if you say that to a medical scientist, they think you're talking about the ability to conceive. So we've adopted this term, reproductive behavior. So what we found in this study was that if we use whole genome data, we also found the SNP heritability. So whole genome heritability is about 15% for age at first birth and 10% for number of children ever born. So this was kind of interesting. We thought, okay, it seems to be when you use whole genome data that there's really a genetic basis to when you have your first child and how many children you have. And demographers and social scientists have been not looking at this at all. So we were quite excited about this. But then you get to the question of, again, it's still that heritability thing where it's like 10 or 15%. So you're kind of wondering, well, so what? So it's 10 or 15%, but what are the actual genes? And do they have a biological function? And I think sociologists are thinking often in terms of mechanisms. So we kind of wanted, we wanted to figure out what's the mechanism. So how is this gene, linked, or these genetic loci linked to particular outcomes of postponing or having a higher or lower number of children? So for this reason, we embarked upon a genetic-wide association study. I thought it would be relatively easy, and then 4 years later, we completed it. So it was quite a massive thing. And what we did is you probably have seen similar talks to this, but it's basically data mining across the genome to see what the relationship is with certain genetic loci with your outcome of interest. The geneticists like to call it hypothesis-free research, but in sociology, we often call it data mining. I've also heard it termed as agnostic statistics, which is another nice one. And I think what's really interesting with this is that you have mandatory sort of independent quality control centers. So we did our analysis in Oxford, and then someone else did the same analysis in Rotterdam, and now we have another group doing it in Cambridge, and then we come together to see if we get the same results, which is really interesting, and I think it's a nice way to do replication. This was a paper that I'm working on now that I was hoping to discuss with Dalton this afternoon, but I might have to fly out, but we'll see. We did an analysis of all the genome-wide association studies to date. There's over 3,000, and we did sort of a data science approach to it to look at what they've done. And I think there's a few sort of striking things that come out, and this is just an aside. You'll see that, these kind of studies of finding genetic loci started in about 2007, but they've increased over time. And this is the sample sizes. So the sample sizes have gotten larger, and actually within the last 2 weeks, they've gotten up to 1 million almost, some of them. And I think what we focus on in more detail in that paper is the ancestry, which Dalton's talked about in his work, but others have as well too. So blue shows those of European ancestry, and Asian is red, and we show the other groups too. So I think it's important to pause and look at these genetic studies and look at the diversity that is required in the future. We go in that in great detail in that paper. But going back to the study that we looked at, so it was published in Nature Genetics in 2016, October 2016, and it's now old, if you can imagine. The field moves very fast. And in that, we identified 12 loci that are influencing age at first birth and number of children ever born. At that point, it was one of the top 10 largest ever GWAS studies to date. So we grouped together over 60 data sets from vastly different areas, and we critique this a little bit later in another paper. And we also, used data from a large American insurance company, from direct-to-consumer data sets, and as well as a lot of scientific data. And I think what's quite exciting is not only the sample size, but that we have a lot of information on men. So a lot of the research on male fertility and infertility, as you know, or anyone that's worked with the Demographic and Health Survey, either the data is not collected for men, or when it's collected, it's from very specific samples of men that are worried about their fertility. So this was really interesting to get this large sample of men. So these are the results. You might have seen these before. They're Manhattan plots. That's the chromosome, and this is significant, so this is suggestive, and this is actual significance. And these are the genetic loci that we isolated for age at first birth and number of children ever born. So these are well, you use all SNPs in your weighted score, but these are the ones that have the strongest weights. And I won't go into this in detail, but then what we do is we return our results over to biologists and molecular biologists to see what's the cause and what are these genes doing. And don't worry, I won't go into all of these things in detail, but you do a lot of sort of different analyses, epigenetic analyses, and different causal analyses to see, what is the function and the role of these genes. What we found was really interesting. In fact, I think the reviewers, and the most excitement came from the male findings, in fact. And we didn't know a lot about, sort of male fertility and infertility. We know something, but, a lot of the things that we found in our follow-up studies are showing that it's related to sperm defects. So men that have later genetic propensity to have children or a lower number of children have a higher incidence of having either low sperm counts or problems with sperm mobility. So this was interesting. And actually, someone's taking our results and using it in relation to IVF treatments, because some of these things can actually be corrected for as well, which is interesting for a sociologist to have it end up there. It's also related to development and also hormones. And as well, what we were seeing is it was related to infertility diseases in women as well. And something that we're following up in a second study, but it came out very strong in this study, is there seems to be some genes have a sort of biological or epigenetic function in the sense that they seem to alter the expression of genes, which is particularly related to lower sperm count and quality. And then we wanted to see if the other, what we were looking at, is it associated with any other genetic traits? And I'm not going to describe all of these, but I just want to give you a flavor of what we've looked at before I turn to the full analysis. So we looked at, this is LD score regression, and it shows the genetic overlap between, in red is age at first birth, and in blue is number of children ever born. And these are also GWAS studies for lots of different traits related to development, BMI, all different sorts of measures. And one I'd like to focus on, and one that we've pursued in more detail, is the genetic loci that we found really clearly appear to be linked to human development, which is quite interesting. So it's related, there seems to be something in relations to hormones, and those that had a genetic propensity to have a later age at first birth also have later menarche, later menopause, and also later age at first sex. So these were kind of interesting things. And for boys, we were able to see that they also had later voice breaking. That was sort of the only measure that we could get for boys that showed some sort of biological development. That's always tricky. So then we started thinking about it, and something that we're going to pursue, but what we were planning to, is it appears that there's a certain group that appears to be shifted, at least in terms of genetics, into all of these developmental phases appears. So menarche, sex, birth, menopause is happening later. So we thought, wow, I wonder if this is related to longevity. Of course, somebody else thought of it and beat us to it, but that's fine. They cited us. They show actually that our age at first birth polygenic score is related to longevity. So we're following that up as well, too. So it's really interesting to think about these different links with fertility and mortality. So something that we're just finalizing now, in fact, I got some results a few hours ago, is we're redoing this study with a larger sample and with different outcomes as well. So we're looking at it, and we're also including childlessness, which is just a variation of number of children ever born. But we saw a high correlation with age at first sex and age at first birth, as you might imagine. But there's a high genetic correlation. So we're also looking at that as well, too. We're including more detailed data, so we have a lot more hits, and we're looking at the X chromosome as well. So the number of observations that we have now is around 800,000, and it's lots of files for our different sort of outcomes. So that should be coming out soon. And just to give you a taste of what we're finding, the last study of age at first sex that found some genetic loci had 38. We're up to over 300 loci that we've identified. And as I just showed you, the old study in October 2016 in Nature Genetics found 12 loci. We're now finding hundreds as well, too. So our biological guys are really busy at the moment. And it's picking up some of the same things in relation to men, but also some other overlaps and that will be coming out soon. So that's the genetic side. So I looked first at the social demographic side in my work, and then I wanted to look at the genetic side, and then I realized, okay, no one's done a genome-wide association search on these outcomes I want to look at, so we did that. And now I want to combine them together, because actually that was, as a demographer in sociology, the thing I wanted to do in the first place. It's nice to have the genetic discoveries, but it's also, I wanted to see if you enter these genetic variables in the models, will they have a high predictive power, or, what happens with gene-environment interaction. So we looked at it to explain childlessness. And a question I get a lot from people is, how on earth could you look at the genetic transmission of childlessness or infertility? So that's the question we get all the time. So we decided to just write an article about it to explain it. And that's published last year in the European Journal of Human Genetics. And one of the main reasons is what the biologists and the geneticists understand right away. It's called sexual dimorphism. And you can look at that paper if you're more interested. But basically what we find and what we empirically show is that there's different sets of genes for childlessness and infertility for men and women. And those can be passed on via the female lineage or the male lineage. So this is the difference. And we're now also looking at intergenerational models where we show mother-to-daughter transmission and father-to-son transmission and cross-transmissions to try to show these things empirically. So that's how it can happen. So you can skip a generation and you can pass it on through the male or female lineage. So looking at the conceptual model, this is the review, and this is what I did for most of my career, looking at the social aspects. And then I just told you about the GWAS, the the behavioral aspects. We're including this in the model. And then what I'm also including is a lot of GWAS studies that have looked at infertility-related traits. And I want to see if this relates to postponement and if it predicts childlessness. So we're also looking at G by E, so gene-environment interaction, is there interactions with cohort and age, and also gene-gene interaction as well. So that sort of gives you an idea about the model, the conceptual model in the paper that I'm going to show you now. So we know that infertility has different causes for men and for women. For women, a lot of the research has been, and most of the research, as we show in our other GWAS paper, most of the research has been done on female infertility factors. I think it's because of the data availability. But in our other paper that I showed you about the GWAS studies, I even looked at the gender of authors. Sorry, I'm interrupting myself in my own presentation, but we looked at the gender of authors, and actually we were surprised to see that women were studying more female traits, such as breast cancer, and men were studying cardiovascular traits. So this is really interesting as well, too. So we looked at the gender of the researchers across time as well. But that's another paper. So what we found is there's a lot of research on female infertility, so we want to include that in the model. There's less on male infertility, and the samples are very small and very selective, but I'll show you that in a moment too. And then we have a lot of unexplained factors. So we're going to be adding in endometriosis, but also genetic-wide studies that have looked at low sperm count or low sperm mobility. We also, there's been a lot of researchers, Dalton being one of them, that talked a lot about differences across cohorts and how we might think that different genetic propensities might be different across different cohorts, or different genes might be important in different time periods. And this has been discussed a lot by Udry, but also others. And this is just a paper that we published a few years ago showing how heritability seems to sort of vary across time. So if you're in a period where you have more choice and more liberalization, perhaps in relation to fertility, the genetic propensities might be more realized. And then just to give you an example, a personal one, this is my mom. And this is when she had her children, and then this is me. Look at how happy and free and no social control in my case. But think about it. If you think about these cases, you have this case. You can see this is the mother-in-law, my grandma, and you can see her mother. You can actually, or her grandmother, or her grandmother. You can see actually the social control in the room. I mean, it's there. It's saying, this is when you're going to have your first child. This is what you're going to wear. This is, so the social control is very, very clear. And when I was deciding, to have children, there was much less social control. So the theory then is that your genetic propensities might be more important in those periods. Whereas in these cohorts, the social control is more important. You can also see the heritability of eyeglass use as well in this picture, which is really clear. This guy doesn't have it yet, but it's coming. Okay. So also we wanted to look at, and this is kind of funny for demographers to think that looking at gene-environment interaction across age, but this is something that has been done much less, actually, in these research. So they haven't looked at cohort or country or age differences. What we thought would be interesting, we know that there's a decrease in the ability to conceive across time for men or for women, and there also is an increase in sperm defects for men. So we wanted to look at these interactions across life course phases as well and age. We also wanted to look at to see, one thing we've been thinking about a lot is a problem that we probably have, and we've shown it in other studies, is that our genetic scores for age at first birth and number of children ever born, are they purely genetic risk scores? Or are they picking up something else? Okay, so are they picking up things that are related to childlessness or related to having higher education or postponing? And, so we remain skeptical about this as well, so we wanted to test it. And we've seen that the genetic influence on number of children ever born is actually quite shared with age at marriage. And also, if we look at the correlations between the genetic, the GWAS study on education and our age at first birth, there's a 0.7% correlation, so genetic overlap in those two traits. So they must be picking up something else that's not only genetic. That would be our assumption as well. So we wanted to test this. And as well, I'll see if I have time, we looked at various gene-gene interactions as well. So the data that we use, which this group is probably familiar with, we replicate it because I find it extremely important with these kind of studies that haven't been done before using the genetic data, that you replicate it using multiple data sets and as comparable as possible to see, okay, will this replicate across various data sets? And I'll show you it replicates in some ways, and in some ways it doesn't, which is interesting as well. So we used the health and retirement study and the Wisconsin longitudinal study that I'm sure people are familiar with. And we select people that have reached the end of their reproductive period because we're looking at childlessness. The measures are the usual suspects that we include for the socio-demographic measures, but we also had to harmonize it across the data sets. So it's age at marriage, educational attainment, occupation, religiosity, or actually it's religious affiliation and birth year. We add in these polygenic scores. I described these already, so I probably don't have to repeat them, but these are the sort of single quantitative measures of genetic risks that are taken from the GWAS studies. So we first added in what we call our behavioral score, our behavioral genetic score. So that's the one I showed you that we published for age at first birth, the number of children ever born. And then we also added in the more biological polygenic scores. So things related to infertility, diseases for women, sperm defects, and mobility problems for men. And then we ran 4 different models, and I'm just going to give you the summary of the final models today. Because as you can imagine, we split it. We ran it for men and women, and we also ran it for the HRS and the WLS, so there's lots of tables. So I'm just going to show you some summary results today. We first looked at the predictive value of the polygenic scores of the behavioral, so age at first birth, the number of children ever born, and then those of the biological phenotypes, or biological GWAS, and then we looked at the social demographic variables, and then we added them all together. It's hard with these, childlessness is the outcome, so 0-1, so it's a logistic regression model. It's kind of hard to find a good R-squared, but we chose just to look at the explanatory power. So, we wanted to see, by incorporating these genetic variables, do we increase the explanation of variance? So have we been missing something all this time? And, so it's the social demographic variables and the genetic variables. What do you increase your explanation of variance if you include them? We also run, I'm not going to go into detail, but separate multi-level models because we have individuals with siblings in the WLS and couples in the HRS. And we do lots of different sex differences, and we look at, to pull out some of these gene, environment, and gene-gene interactions, we do interactions. So to assess the confounding, I think many people know you can't compare logistic regression odds across models because of unobserved heterogeneity. So my colleague, Richard Breen, who's going to be coming in a couple of weeks, I think, together with others, developed this model. So that's the way that we're going to look at, and we're sort of assessing the percentage of confounding due to the fact, remember I said before that our polygenic scores for age at first birth and number of children ever born might be picking up some unobserved factors related to those that are childless. So they might have, because they have higher education or those kind of factors. So we want to see what's the percent of confounding. So we first sort of always like to do some descriptives and compared with the human fertility database what the levels of childlessness are in the HRS and WLS. We see it's slightly lower in the Wisconsin longitudinal study, and it's a different sample taken at this period. So, but it shows that they're roughly similar across time with some variation. And then just moving to the results. So what was really interesting, I kind of love this topic because you go into it, at least as a social demographer, sort of wondering, what's going to be more important? Well, I knew that the genetic scores wouldn't be that high of predictors, but I did think that the biological scores that picked up infertility would be better predictors of childlessness than are ones that we measured, the age at first birth and number of children ever born. But that wasn't the case, and I'll return to that at the end. So our polygenic scores, and I'll show you some of them in a minute, for reproductive behavior were actually the better predictors, and the ones that picked up the more biological aspects turned out to be not very good predictors of infertility or not having children. The effect of social factors was as expected, and I'll show you that. And then if we enter these variables alone in the models, the polygenic scores, so genetics explains about 1 to 2% of the variance. And I think that's, so we don't need to quit being sociologists and demographers looking at these topics. I think you already realize that, but it's nice to put it into perspective. And then the social factors, well, they're better, but in the HRS they explained around 19% and 46% in the Wisconsin longitudinal study. So remember that we have limited social factors for comparability. And if you actually start looking at the R-squared models in a lot of demography and sociology articles, you'll see they're actually quite low. So I just want to say that as a caveat. And what's more interesting is if you include everything together. So you can increase your explanation of variance, but don't get too excited by it, because it still should remain low. And that's kind of what we would expect for these sort of complex outcomes about deciding when you're going to have a child or how many children you have. If it was, as some of the twin studies said, 40% heritable, that would almost be hard to believe, With a caveat, I have to say, in our newer study, we're getting up to around 4%. Predicted variability. So that's the new one that will be coming out. So it's not, if you enter in education alone, it's in some of the models maybe as a univariate 12%. So it's still something sizable, but it's not going to overturn what we know about fertility and demography. Okay, so I just graphed some of the results. I realized it's nicer than looking at the tables. This is for childlessness. Remember, that's our outcome for the HRS. Results are broadly similar for the WLS, but a bit stronger in terms of significance. You see that our polygenic score, so the genetic propensity to have children later significantly predicts childlessness. And as well, we see things related to birth cohort education. If you marry later, you have a higher propensity to be childless. So things that we know already. We already know a lot about the employment literature compared to clerks, which is the reference group, women that are managers or professionals. We know from the literature in certain occupations have lower levels of having children as well. And for men, with the WLS, actually our age at first birth polygenic score was significant, but it wasn't with HRS, so I'm showing you the results for HRS. And actually not much was going on there, and I'll return to that just when I conclude. So the genetic scores that are supposed to measure infertility and fertility problems are not predicting childlessness very well. So that just summarizes that. And we looked, so birth cohort education didn't seem to play a role for men, so we know this from other studies as well too, also in the WLS. Age at marriage was the really strong predictor, and of course in all of these, I'm not showing you this, never having a partner was the strongest predictor of being childless. Yeah, so sorry, I should have said that. So, and particularly for men. So whereas higher education was for women, we know this, but for men it was not partnering. And of course, occupation didn't matter, which we know from other studies as well. So what was interesting, particularly if I just show you some results, is the gene-environment interaction with age. It's not a very pretty graph, but if you look at, these are the polygenic scores, and this shows that you have a higher genetic propensity to have a later age at first birth. And we see that the slope is actually, for the women that enter into marriage later that are probably then trying to conceive at that time if they're haven't had children, we see that actually has a more important effect. So this is really important, I think, for demographers and sociologists to come into this kind of research. Will Dalton has argued this quite a few times before too, that you have to take a life course approach, because different genetic factors might be more important as people age, and this is definitely the case with these fertility-related factors. And if we look at other factors, it's what we would expect. So our polygenic scores for later age at first birth, so you're genetically prone to have later children or a lower number of children, is definitely related to higher education and later age at first marriage. So it's what we anticipated, and we can see that in HRS and WLS broadly for men and women, and it's yellow is significant. If we look at the G by G correlation, so our genes correlated with each other, we see that there's a positive correlation with age at first birth, genetic propensity, and endometriosis, but nothing really happening for the male studies, and I'll return to that in a moment. So was it all confounding? So the polygenic scores that I worked for 4 years to produce, the age at first birth and the number of children ever born, are they actually genetic scores, or are they picking up other confounding factors? Well, when we enter it into our model, and I have the tables if you want to look at it after, but I just summarize it here. The effects for age at first birth and number of children ever born, those genetic scores decrease by about 25 to 85% if we include education and age at first marriage. So it looks like our polygenic scores are really capturing, as we thought, these factors that are linked to childlessness. So things related to higher education, entering into marriage later. And there might be a pleiotropic effect, so genes that are influencing those to go into higher education, but also postponement. We might have effects going on there, so we're going to explore that too. And the biological polygenic scores didn't do much in any of our models. So I wanted to leave some time for discussion too. So just to conclude, it looks like at least the polygenic score for age at first birth is interesting, and this is the old polygenic score, but the new one is predicting, as I said, much higher levels. Age at first sex turns out to be a very strong predictor for multiple outcomes. So we're going to make the genetic scores available, the day we publish, and we're going to link it up to as many data sets as possible so people can use them. So I think it'll be really interesting. There's high correlations with smoking, risk behavior, development, and we seem to be picking up a lot with this age at first sex variable. But again, it could be influenced by risk behavior or other factors as well. Contrary to what we thought, our biological polygenic scores actually had very little explanatory power. When you look at them, they were actually done from quite small samples, and particularly for the men, it was data collected once you look at the samples from fertility clinics. So people that were worried about their fertility, and they were small samples, so there's probably something related to selectivity there and just not having enough power. So I guess it was a win-win situation for us. We thought it was interesting to look at the genetic effects, but the social factors, as we would anticipate, still play a strong role. And I think what's really interesting, and something we're all pursuing, I think, in more detail, is it appears that some of the genetic factors related to infertility, particularly I was looking at it in detail with endometriosis, they only become relevant or are triggered by certain ages. So once you get older, you might have this genetic propensity to develop, sort of diseases related to infertility, but they kick in at an older age, which is only relevant when people postpone. So that's why we're only seeing these effects now as well. So higher education, I talked about that in age at marriage. It's correlated very much with these variables, so it'd be interesting to sort that out in more detail. And I talked about pleiotropy as well. So I think we're going to now look more at why is there this huge overlap between the GWAS for educational attainment that I think maybe Dan Benjamin might have talked about, and is a 0.7 correlation with the age at first birth variable. So what's being measured here genetically. But I think it shows that it's interesting to combine these things. We can increase our explanation, and I think it's good to put it into perspective and to include these variables in similar models as others have done. Some caveats. Some of the results from the samples differed, and that's why I love replicating things on multiple samples. We didn't find the age effect in the Wisconsin longitudinal study, so the genetic effects didn't kick in for older women. They weren't resulting in higher levels of childlessness. But once you look at the sample, there's a reason. There was a very low number of women that actually married at older ages. So it's related to a sample and a substantive reason. I think some of our GWAS also, the biological ones, just had very low power. And I think anyone going into this field, and other people have written about it, is when you're looking at this gene-environment interaction, you have to have large samples. But something that we've worked on more recently is, and this is maybe funny for demographers, so when we first started working on the GWAS, we combined 63 different data sets, and they came from multiple cohorts and multiple life phases, and they came from a whole bunch of different countries. And I kept saying, like, putting my hand up in the lab meetings, like, with the geneticists, I was like, this is a problem, isn't it, for heterogeneity? And, you know, that they're from different cohorts and different countries, and it was just a blank look, like, who is that? Who let her in? So Dalton's written about this before, too, but we've now finally empirically shown that you actually, you know, have heterogeneity and hidden heritability when you don't look at the countries that people come from, so the populations where they come from, and the cohorts. And I know it's sort of laughable to a demographic audience that we're introducing age, cohort, period, and country into these models, but you know, it's important to show. So what we show in this study, and I think Felix was here last week too, a postdoc of mine, is, you know, we show that you actually miss a lot of the genetic explanation if you don't take into account the country that people come from in the cohort. It's very basic, I realize, for this audience, but it was something that we were able, and it got to the cover of Nature Human Behavior, so adding in cohort and country into the analyses. So that's what I wanted to talk about. We're not very good at updating the web page, but we'll try. So we put our publications, and all of our results are available. And soon we're putting all the code that we use to do our analyses also, making it all open science. And so that's going to be on Socio-genome and as well on my webpage. So thank you very much.
Okay, we begin with a question from a graduate student.
Hi.